How can I tell if any of these plants that I see are actually colonized by mycorrhizal fungi? The answer is you can't tell by just looking at that plant. It might, for example, be smaller or larger for any number of reasons that have nothing to do with mycorrhizal fungi or even the soil. Well, and even if you were to take a plant and dig out its roots and look at the roots, you wouldn't be able to tell either, because there's basically not much of a difference between roots that are colonized by at least arbuscular mycorrhizal fungi and other root systems. That situation is a little bit different for ectomycorrhizal fungi, but maybe we'll talk about that in another video. Let's talk here about arbuscular mycorrhizal fungi, one of the main type of mycorrhizal fungi, important in grasslands, many tropical forests, and also in many crop plants. So how can you tell if any plant here is colonized by mycorrhizal fungi? Well, the answer is not that simple. You have to dig out that root and you have to take it back to the lab. Back in the lab, you have to treat that root with a bunch of different solutions. First, you have to clear the roots with KOH to get rid of some of the cellular contents, and then you put it in a acidifying solution, and then you put it in a staining solution. The stain that's used is usually trypan blue, for no particular reason other than that it works, or sometimes you can also use black ink. After you've done that, you need to mount the little tiny root pieces after the staining on slides, and then you have to take a cover slip and press these roots down to flatten them out. So it's basically a squash preparation. And after you've done that, you have to look at it under the microscope. Pretty high magnification. We usually use 200 times magnification. And then you can see the structures characteristic of arbuscular mycorrhizal fungi inside of the roots. What people then usually do is they go through that slide under the microscope at regular intervals, usually look at about 100 of these intersections, and at each intersection, the microscope crosshair, we count what structures we see. And then in the end you get a value that's percent colonization, for example, by hyphae. And this is the values that you often find reported in the literature. Beyond just are these plants mycorrhizal or are they colonized at all, you also get this quantitative estimate of how much are these roots colonized by arbuscular mycorrhizal fungi. Yeah, so as you can tell, it's not that simple. It requires quite a bit of lab work. Maybe in the future there's going to be other methods available that are based on some biomolecules that these fungi produce or their DNA. There's also some other methods available based on phospholipid fatty acids, but they're even more complicated and also much more expensive. So the method is not ideal, but it's widely used, and this is how you find out if your plant is colonized by arbuscular mycorrhizal fungi. With that, see you next time. Bye.